Okay, on this video we're going to talk about another example of vesicle and vesicle traffic, moving vesicles, and it is this example of receptor-mediated endocytosis. And so your book describes that, and the example we're going to talk about is bringing cholesterol into cells. And our body can synthesize cholesterol. It's often done in the liver. And let's start out by taking a look at cholesterol and seeing what the molecule looks like. Here's a couple of drawings of cholesterol. Cholesterol has these carbons, and you can see that back to sort of chapter 2 that, well, this isn't completely drawn. Here's a carbon which is one covalent bond. And when carbons don't have other bonds formed in biological chemistry, it's assumed that these would be just hydrogens. So what they've done is they haven't drawn in lots and lots of hydrogens here, but let's just draw in some of them. And the reason they haven't is because say for example here, they drew in this OH group. It's just understood that carbon's going to have 4 bonds, and if it's not shown, it must have another bond with hydrogen, just because that's sort of a simple, unreactive, non-polar covalent bond that carbon could have. So here's another way of drawing it where they don't even show the carbons. See, it's the same molecule. They're not even showing the carbons in this part of the chain, and that's you often see this drawing where there'll be a ring structure in a molecular model, and it doesn't show the carbons or the hydrogens. And so you can take a look at this, and if you imagine putting in the hydrogens, this cholesterol is very hydrophobic. So all of these are non-polar covalent bonds. Cholesterol is like a fat or an oil. It just would not dissolve well in water at all. Here's the only hydrophilic part of the molecule, just a tiny amount, like that. Our bodies can synthesize cholesterol. We have it in our diet, and where that work gets done, whether it's digesting cholesterol or synthesizing cholesterol, is really typically in liver cells. And where would it be in liver cells? It'd be in the smooth ER. And that's where cholesterol is made. And elsewhere in your body, it's not really made much, but cells need it. And for example, this is the starting point for synthesizing our hormones estrogen and testosterone, which gets synthesized in the ovary and testes. Testosterone. And so we need to get some of these building blocks to cells like the gonads, ovaries, and testes, where they're used to build other molecules. Also, there's lots of cholesterol in all cell membranes, so cells need cholesterol for a variety of different reasons. So often organized in the liver and then released into the blood, but it just can't be released as cholesterol because it would not dissolve in the water environment of the blood at all. So here's what happens. That cholesterol in the liver is packaged into particles, and one particle is called low-density lipoprotein, LDL, and here's what LDL looks like. The yellow is cholesterol that is packed in a hydrophobic core surrounded by, see these phospholipids? So it's a little bit like a micelle type structure. It's not a phospholipid bilayer. It's just the heads on the surface to make a hydrophilic, water-loving surface of this LDL, and tails into the interior that's very hydrophobic. So a hydrophobic interior containing cholesterol, and it's going to be delivered because it's going to be soluble in the blood, like that. And one other component is there's a protein, and the protein is going to provide a handle. So when a cell wants to get cholesterol, this is going to provide a handle for the cell to grab LDL and bring it into the cell. And the way it's going to do that is there's a complementary surface on a protein called the Ldl receptor. And the Ldl receptor is a membrane-bound protein. So here's aquaporin in the membrane. Something else we talked about was this cystic fibrosis channel might be in the membrane, like that. in certain cell types. And then here's the LDL receptor. It's in the cell membrane, and it can bind through non-covalent interactions with the protein that's part of LDL. It'll bind it, and then this material is going to be brought into the cell by making a pit, and then pinch it off and form a vesicle containing LDL, and containing LDL because it is bound to the LDL receptor. So it's a great example of this story of receptor-mediated endocytosis. We're bringing stuff into the cell, and we're bringing the cell this stuff the cell wants because the cell has protein receptors that bind to that material, and then it gets endocytosed into these vesicles. Okay, so let's talk a little bit more about it. And let's go back to here and just again say this is what we're talking about. An example of endocytosis called receptor-mediated endocytosis. And what happens is that the it's back to here. See, it's to be contrasted a little bit with pinocytosis, where pinocytosis is just kind of randomly grabbing stuff out of the extracellular environment and bringing it into the cell. And here we're selectively grabbing stuff out of the cell, extracellular environment, in this case the blood, grabbing some LDL because this cell wants LDL. It might be a cell in the ovary or testes that wants some cholesterol to build a steroid hormone like estrogen or testosterone. More typically, it's any cell, because any cell needs some cholesterol, and it'll be grabbing it and pulling it in. Okay, and so what happens is that these receptors, when bound, change their shape. And so let's just draw a receptor in the cell membrane, and here we'll say this is the shape of a receptor when it's not bound, like that. Okay, so this is the LDL receptor protein, and we could go back and we could talk all about it. Hey, do you think the LDL receptor protein has a signal sequence when it's being synthesized? The answer is yes, because it needs to be delivered to where it does its job on the cell membrane. And so when it's getting made, it's a long chain of primary amino acid sequence that's got some secondary sequence, some twists and folds to it, that folds up into a three-dimensional tertiary sequence structure like this. And what happens is when LDL protein changes its shape, binds to LDL, the LDL protein changes its shape. It wraps, it's kind of like a pair of pliers, where the shape changes here as it kind of closes around the LDL. And as a result, there's a conformation change here, like that. So the LDL receptor goes through a conformation change upon binding. And I mentioned briefly in class that that's common for tools. Like this is a little bit like a pliers, where part of the function of the pliers is to be able to change shape in order to do its job. And that's different than aquaporin, which is just a water channel, and it's just a stiff water channel that's always open. There are other channels that open and close that go through these conformation changes, like a tool that changes its shape to its job. And as a result, they expose binding sites here in the cytoplasm that allow the bound receptor to start to cluster. So we're going to selectively pull bound receptor, LDL containing receptor, into the cell because the shape change allows the interior of the cell to identify this and cause it to cluster. As it's clustering, that's helping build this this vesicle. So we start out with a smooth membrane, and then bound receptor starts to cluster, and as a result, the membrane starts to bend in here. Now, why does it start to bend in here? It starts to produce what's called a coated pit, and it's called a coated pit because under electron microscopes, scientists observed that the beginning of endocytosis and the formation of vesicle included proteins binding to this surface. So we've got LDL receptor clustering, and then at these tails, there are proteins that are starting to bind at these tails, and the protein that starts to bind on these tails and it causes it to form this coated pit is our next protein to know about, and it's called clathrin. There it is, clathrin. The coat of a coated pit for here is called clathrin. There are, it's a thought that all vesicles, when they form, wherever they form, the formation process includes a coat protein, and there are a variety of different coat proteins, and we're learning about one, clathrin. And so another part of the cell's story is that that vesicles are first formed by having these coated vesicles form, and then the coat is removed. So let's just add that. How's that happening? Well, it turns out that if we look at the structure of clathrin, structure gives function, and clathrin is a protein that has quaternary structure also. So if we just think about clathrin, clathrin's a protein that looks like this, has a shape, and it has a couple of binding sites on it, like that dents that allow it to bind to other surfaces. And clathrin has a primary amino acid sequence. So it's a chain of amino acids joined together by covalent bonds. So if we zoomed in here, we'd actually find a chain, and it has some local twists that give it a secondary structure. And then it folds up overall into a three-dimensional tertiary structure. And clathrin doesn't look like a donut. Clathrin looks a little bit like an arm that has a hand at one end and a hand at the other end. And clathrin also has quaternary structure because when clathrin assembles as a coat, it actually assembles, and this is a little bit hard to draw, so I'm going to try to draw it over here. It actually assembles as a three-component protein, and that's its quaternary structure. Quaternary means that this protein to function binds to more than one other protein. In this case, it binds to 3 clathrin proteins, and those start to form a pit. The reason that's significant is because the clathrin, when they are bound by their hands at the base, forms like a puckered triangle. So I'm trying to draw that a little bit. Maybe I could draw it out like that, where this is like a puckered triangle, and that's what helps create the dent, is that as the coat gets added, the coat clathrin proteins are puckered or bent like this, and just by their assembly on this surface, they start to form a dent. Now, why does clathrin form here? Part of the reason is because there are receptors clustering, and part of the reason is because other proteins are also assembling here, helping guide clathrin to build a coated pit that will then, as more and more clathrin builds up, form a coated vesicle and pinch off from the membrane. And why is this happening? It's happening spontaneously, and this is one of our first examples where it takes energy. So we're going to find that energy is used, some of the energy of the calories you digest is used to help these vesicles form and pinch off like that. So clathrin pinches off, and in doing so creates a coated vesicle inside the cell. The coat gets removed, and we have a vesicle. And the vesicle has the receptor with LDL bound to it. So we brought some cholesterol containing LDL in the lumen of this vesicle into the cell, and now what happens is that the receptor gets released from the LDL, and the LDL is delivered to the lysosome. So the lysosome is going to digest and break up the LDL and then send some of it to where? It's going to send it to the smooth ER, where that cholesterol is going to be metabolized. It may be synthesized into modified forms like steroid hormones and so forth. The receptor gets recycled back to the cell membrane. So here we have unbound receptor heading back to the cell membrane, where through exocytosis it's delivered back to the surface of the cell. And that's that. So we have a nice cycling process here of coating and pulling in bound receptor, and then releasing the coat, releasing the sort of the baggage, the LDL, and sending the receptor back to the cell surface. The reason this comes up is because LDL is important in health, and sometimes people have high levels of cholesterol in their blood, and sometimes people have high levels of cholesterol in their blood because they inherited problems with digesting and using cholesterol. And here's an example where that could be disrupted. The person could have inherited a DNA that has a problem coding for the LDL receptor itself. And if there's a defect in the LDL receptor, they're not going to be pulling cholesterol into the cell as well. But then in this pathway, you can see there are actually several other players that could also have a problem and lead to higher cholesterol. There could be a problem with the Clathrin. There could be a problem with the other proteins that assemble to form the coated vesicle. There could be a problem with the proteins that are involved in uncoating the vesicle. And so all along this pathway, there are several proteins involved, and we'll learn more about pathways like this and how some people will have inherited problems with the proteins that play essential roles along these pathways. And so that is that. So we're done with that one. Ta-da!